one I will present. Now it's francielle. We established an in vitro model of vascular permeability evaluation tested hemorrhagic dengue fever pathogenesis and screening of compounds in therapeutic foundation. So francielle is a new postdoc here at the institute, and she just got the project. So what you are going to see is the explanation of this new project that we are doing together with Camila. So thank you for accepting.
Okay. Thank you, Professor Esther, for presenting me. Today, as she said, I'm going to present my postdoc project developed here at Institute of Tropical Medicine, University of São Paulo, under supervision of Dr. Esther Sabino and Camila Romano. This study aims to establish an in vitro model of vascular permeability evaluation to study the hemorrhagic dengue fever pathogenesis and to screen compounds with therapeutic potential. I'll start by giving a brief background information. Dengue is a mosquito-borne viral infection, and after being bitten by an infected mosquito, individuals may present subclinical infections or present a range of symptoms with different degrees of severity, and depending on many factors such as viral and host factors, the clinical presentation of the disease can vary. For example, you have dengue fever in moderate case, dengue hemorrhagic fever, and dengue shock syndrome in the severe case. This classification is very complicated. In 2009, the World Health Organization suggested a new classification for dengue based on warning signs. So they suggest 3 types of dengue: without warning signs, with warning signs, and severe dengue. Although the disease may present a range of symptoms and clinical signs, a hallmark of the disease is the increase in vascular permeability, and this results in plasma leakage and is also related to hemorrhage. This is a cause of morbidity and mortality. For example, about 50 million infections that occur per year. 500 are severe cases and causing 22,000 deaths a year, especially in children. After the subcutaneous injections of the virus into the skin, the virus particles infect the local immune system. It starts predominantly the dendritic cells, and after this first infection, the virus spreads through the lymphatic system and then bloodstream, and getting some other tissues like liver, spleen, kidneys, and then establishing a systemic infection. In the bloodstream, the monocytes are the main infected cells, and it's thought that these cells play a central role in dengue pathogenesis, especially because the interaction with the other cells, lymphocytes, natural killers, and also because the secretion of lots of cytokines, and these molecules interact with the endothelial cells and blood vessels, causing opening the junctions between the intercellular junctions and causing increasing vascular permeability and then plasma leakage. There are few good models to study dengue pathogenesis, and there is some authors that have published some in vitro models to study vascular permeability, and those are based on using human umbilical vein endothelial cells, HUVECs, and these cells cultivated in transwell inserts. These are commercial wells where we have an upper compartment and a lower compartment separated by a microporous membrane, and then putting a gelatin layer on this membrane and cultivating endothelial cells. We can mimic the endothelial layer of cells in the basement membrane, and by adding fluorescent compounds, for example, in the apical side, we can measure the transport of this molecule and then measure the vascular permeability in vitro. Based on these previous works, we decided to establish this model in our lab, but with some modifications. For example, by adding monocytes in co-culture, because those cells are important in dengue pathogenesis and the interaction with these cells with endothelial cells. So we are going to try to cultivate in the upper side with direct contact and in the lower side with no direct contact with the endothelial cells, and compare all the systems. Therefore, the aim of this study was to develop an in vitro system involving the co-culture of endothelial cells in human monocytes, which will be infected by dengue virus, to study pathogenic factors and also to screen compounds which may interfere with the vascular permeability. To achieve this objective, we will have 3 steps in this work. First of all, we will study the ideal parameters in the co-culture system. This is a picture of the real transwell inserts in a 24 well plate. We choose different from the previous studies, HUVEC, that use HUVEC. We choose the human lung microvascular endothelial cells because we believe the cells may be more related with the dengue pathogenesis. Those cells were purchased from Lonza. And also human monocytes. We decided to choose this THP1. It's a cell line from ATCC cell bank. And looking at your presentation, maybe I have an idea to have to use PBMCs also. This table then summarizes some parameters we're gonna test. So the endothelial cells alone and together with the THP1, different cell concentrations, the 2 different cell co-culture systems. We also will test different MOIs and different strains of the virus. We have the 4 serotypes, but firstly we will test the 2 strains from dengue serotype 2. The cell monolayer must have a complete confluent monolayer, so we need to check this every day during the culture by microscopy and also monitoring these parameters, transendothelial electrical resistance, TEER, and by measuring the TEER we can evaluate the monolayer integrity. We will also characterize the monolayer by using some specific markers for endothelial cells. For example, this is a protein, V-cadherin. It's a transmembrane protein which participate in the cell-to-cell junctions. And by using immunofluorescence staining, we can look at the cell organization in the system here we can see the expression of VE cadherin by using specific antibodies. And also in this confocal microscopy, we can see if it's a real monolayer, well organized, differently from here, that in which we don't have an organized culture of endothelial cells. The second step of this study is to compare the in vivo cytokines profiles, comparing data from infected patients. This data has already been collected and published this year. So cytokines were measured from 132 patients during the outbreak in 2010 in São Paulo. So we have the profile of these patients, including patients with dengue with and without warning signs, and then measuring these cytokines in our co-culture system. In the supernatant of the culture, we can compare the in vivo and the in vitro system and see how they are comparable. The third step of this study, once we have the model set up, we can test different compounds to see if they can interfere with the increase in vascular permeability caused by dengue infection in vitro. We choose this compound, ecin, which is a natural compound extracted from seeds of horse chestnut. It's already used clinically as an anti-inflammatory, anti-edema, and capillary protective. Also, some studies have shown that ecin reduces TNF-alpha and interleukin-1 secretion by monocytes. And we will also test this polysaccharide, which is a glucomannan, here's the structure, and is extracted from Agaricus brasiliensis, which is a mushroom native from Brazilian biota. And this mushroom, it's very well known to its medicinal properties, especially related to immune regulatory activities. So we believe that these two compounds may interact and have some activity in our model based on traditional use. Unfortunately, I still don't have results. As Professora Esther Sabino told, we just started the project. We are still ordering supplies and producing virus stocks. Just to illustrate, here we have the CC36 cells, the cells we use to produce virus stocks. On the left, the uninfected cells, and on the right, the infected cells. Clearly, we can see the multinucleated giant cells as the characteristic cytopathic effect. With this study, we expect that we can establish the methodology for evaluating the vascular permeability in-house. We also expect to find promising compounds that reduce vascular leakage caused by dengue, and these compounds may have some applicability for dengue treatment. And we also expect to be able to conduct studies on severe dengue pathogenesis by testing different serotypes, genotypes, and levels of viremia. And also we can look at antibody-dependent enhancement in vitro. With that, I would like to thank CNPq for the postdoc fellowship. I would like to thank my advisors, and thank you for the attention. I can get questions or suggestions.
So I think it's a great idea looking at the permeability, and that's really important. I wonder, at our institute, there's someone interested in looking at that for brain injury, traumatic brain injury, and
there
trauma.
There is a model for blood-brain
Yeah, so it's,
permeability. It's very used.
Similar.
They use co-culture also.
and what she's found is that by adding plasma or platelets to the system, it actually helps the cells get back together, the endothelial cells. So thinking about how that might apply to dengue, I mean, you have thrombocytopenia in severe dengue often. So I wonder if you can bring platelets into your model, adding platelets or not, to see if that would...
Yeah, I haven't thought about it, but probably because it's a similar system, just the cell is different.
Yeah, just putting one more thing in.
Probably I'll have to use anticoagulant, I don't know.
Yeah, but that's something I thought might have real
because
implications.
in vivo, it happens some recovery after the infection. The permeability is recovered. There is not much damage in this endothelial cells. So this
Right, it's just like they pull apart and then come back together. So can we figure out how to put them back together?
Yeah.
And then I guess you have two compounds you're going to look at. And so if you get the system up, can you make it higher throughput where you can screen,
I don't know how many compounds we'll be able to test but it's not, the median it's expensive, but the cells, it takes like 2 or 3 days to get ready to test, grow. It's like very delicate process. You have to be really careful. I believe I propose like 2 compounds because just to start, but probably will test much more, but I don't know how to prove. So maybe.
When you say the medium is expensive, is the medium with the compound inside, or
no?
it's just
To
pure
grow
medium?
these cells, it's really complex. The medium, there are lots of growth factors you have to add in. They are expensive. They are recombinant proteins. They're really expensive, much more than the regular medium. It's not just serum in medium. You have to add lots of. And also the transwell are also expensive. So
before you get to the screening part, as Philip mentioned, Philip included the third part, but in the third, you have 2 compounds now. You have those monocytes that are infected, and then you're looking at the effect on the endothelial cells.
yes.
So the first compartment is going to be a little bit difficult because I think including more PBMCs and also different factors that might be in circulation in the blood or wherever. You have those monocytes that are infected. It triggers something in the monocytes to secrete some factors and to impact other T cells who in turn are activated and secreting different components. So maybe keeping all the PBMC together, you might get more of those cytokines.
Yeah, probably.
And one way also to check, what you have in your medium, in your culture medium, you could do, maybe we could help you some Luminex multiplex
I haven't
assay
thought,
to
but
see what
we already in that paper they measured the patient, the serum was with this kit from Luminex, but the in vitro gene, I don't remember.
Yes, there's different kits anyway.
It measured like 30 cytokines like at the same time, and we used the same kit to test the supernatant.
Okay, so you're going to do that already.
But as you told, the GHP1, I choose this because it's a cell line and it's good to have the variation, but in vitro there is some pros and cons. So we can compare both and see which one works best.
If you have one cell line, you have a more control mode.
More control
If
mode.
you have
But I've read that GHP1 they don't secrete like much cytokine as the
because what is the assumption for vascular permeabilization? I'm not aware of anything, so it's not my expertise. But I would expect some level of inflammation.
Yeah, it's like inflammatory response, lots of cytokines in activate the cells. The endothelial cells also secrete some markers, some molecules, some cytokines, and it changes the cell in
So,
some way.
so then the second question is, on the second partner, the endothelial cell, would you do some kind of RNA profiling or to see what is the impact before it kind of disaggregates and
Yeah.
become loose?
What we can like do the experiment, and we'll have different wells and test different parameters in different ways, and we can look at the expression, RNA expression by PCR, or looking at the cytokines, and also that using antibodies, we can see how the organization of the cells in the culture after the experiment, like under the infection.